Good afternoon, everyone, and thank you for attending RRS's webinar, Dr. Graciela Ardrani on organs on chips. My name is Cassandra Carlson, and I will be hosting the webinar on the back end. Attendees are encouraged to submit questions using the Q&A box. Questions will be addressed at the end of the presentation. Before I introduce tonight's speaker, I wanted to briefly mention RRS's upcoming events. We'll be hosting a webinar on July 15th on advancing the adverse outcome pathway approach in radiation research through global coordination with Dr. Vinita Chuan and Julie Burt. Don't forget to register for the annual international meeting in San Juan, Puerto Rico on October 3rd through 6th, 2021. Now I would like to introduce our moderator, Dr. Molly Ariane-Keel. In her current position with the Radiation Oncology Branch, National Cancer Institute, Dr. Ariane-Keel leads multiple research projects under interagency agreement funded by National Institutes of Allergy and Infectious Diseases and Biomedical Advanced Research and Development Authority. Her research focuses on using coding and non-coding RNAs as biomarkers to assess radiation injury in individuals who may be accidentally or intentionally exposed to radiation. To bridge the gap between animal models and static in vitro cell cultures, she was instrumental in using microfluidic cell cultures known as human organs on a chip model for studying radiation injury using RNA biomarkers. By recapitulating key radiation-induced responses seen in humans, Organs on Chips provides a more advanced in vitro model for studying radiation-induced normal tissue injury. She is also skilled in analyzing radiation and drug-induced molecular and biomedical changes in cancer cells and normal cells. She is widely published and is a regular speaker at national and international radiation research-related conferences. She has been an active member of the Radiation Research Society since 2005, where she regularly presents her research findings, organizes symposia, speaks on panels, and leads topical review sessions. And I'll hand it over to Molly to introduce Dr. Graciela.
Good afternoon, all of you. I'm extremely delighted today to introduce our today's speaker, Dr. Graciela, because she's an outstanding scientist of the emulate. I was being in touch with her for the last 1 year, like every week. She is advising us how to correct our system. She is acting as a liaison, major scientific liaison officer for the emulate and for the her clients. So she received her PhD in genetics from Albert Einstein College of Medicine. She's been working with the emulate for the last more than 1 year, and she will give you a very good overview today. And I am sure that no matter what is your background with the organ on a chip technology, she's going to give you a very good overview today. Being said that, let me share some of the slides with you, which I am working on the human emulate system. Quickly, I will give an overview right now. We started working on organ on a chip. First, we started with the liver on a chip. Later on, right now, we are working on the lung on a chip. For both these projects, we started as usual in a 2D culture, and later on, we moved to mice, mini pig, NHP. Now we are in a transitional period, which we are checking these RNA biomarkers in an organ on a chip model. And right now we are checking circular RNA, long non-coding RNA, microRNA, and we are checking already diagnosed in the other animal model. Let me give you a quick overview. I'm sure Graciela is going to give you a clear-cut difference between the chip stabilization. After stabilization, we radiate these models. And let me show you quickly how. So this is their chip on a model. Here you can see the pod. This is in our irradiator. We line them in the middle, and then you can see a center line of radiation field. Center line is aligned with channels where the cells are grown. So here you can see liver sinusoidal endothelial cells and hepatocytes in the 2 layer. you will get a clear picture after Graciela's presentation. Our first and foremost question is, when we radiated, did we see some of these genes which we have seen in earlier? Quickly, we checked, we have seen, like, first our question was whether CDKN1 was there. You can see a dose responsive upregulation in the hepatocytes, and in the sinusoidal endothelial cells, major genes were downregulated. Right now, we are checking in various mitigation agents in the liver model. So being said that, I'm handing over to Graciela, which she's going to give you a clear overview. Thank you.
Thank you so much, Molly, for the nice introduction. Thank you everyone for being here today. I am very excited about this opportunity to talk to you today about our human emulation system. And I'm gonna give you an overview of different models of organs on chip. So actually, just as an agenda, so we're gonna start with an overview of our system so you guys understand how the technology works. Then we're gonna dive into a paper from the Wyss Institute in which they actually use the organs on chips for modeling the radiation-induced injury and countermeasure drug responses. Then we're gonna go through some data highlights on different models that we've worked on. And we're gonna close with a conclusion and discussion on the future of organs on chips. So as you probably are aware, the preclinical models that we currently use don't really predict the human response, right? So either because they are very simplistic, which is the case of cultured cell lines, they might be human, but they lack highly differentiated structures. You know, a lot of times it's very hard to keep these cultures for a long time. We also have spheroids and organoids, which have multicellular lineage. They might have some cytoarchitecture that is similar to in vivo, but still they have no flow, they have no mechanical forces, and it's also very hard to perform perfusion studies on those. And animal models, they are very complex, however, they are a different species, right? So that makes it very hard for us to study things like virus or specific diseases, as well as the fact that, raising animals and having these long animal experiments are very expensive. So with organs on chips, what we're trying to do is to recapitulate the organ level physiology of the human body by actually culturing the cells in a dynamic system that have a higher complexity, and that would enable us insight into mechanisms of disease, mechanism of action of drugs, and so forth. So let's take a look at our chip. So this is our chip. It has two channels. The top channel is 1 millimeter by 1 millimeter. The bottom channel is 1 millimeter by 0.2. And they are separated by a porous membrane that have 7 micron pores, which allows the cells to communicate. And we can seed, normally we seed epithelial cells on the top channel and endothelial cells on the bottom channel. And on the side, we have vacuum ports that allows us to stretch the chips, such, for example, to emulate breathing motions or peristalsis on the intestine. And we have separate control of both of these channels, so we can actually expose the cells not only to their respective media, but also to specific compounds or drugs or metabolites. So ultimately, as I mentioned, we are trying to recreate the microdynamic environment of the human body. So one of our features is the tissue-to-tissue interface. So as I just showed you, we have these two channels, which for most of our models, we have an epithelial to endothelial tissue interface, and those channels are independent. And our chip is cell agnostic. That means that we use the same chip for all our models. So basically what's gonna change, it's gonna be the extracellular matrix that you're coding these channels, as well as the cells that you put on. That's what really is gonna make your model. On the bottom here, this is just to give you an idea of the different flavors that we can actually work with these organs on chips. So you can use primary cells, iPSCs, organoids as your cell source. You can work with different species as well as different states, right? So healthy disease or genetically modified. And this ECM also can be different, right? So we can have just a very thin coating of these channels, or actually have a full gel. And I'm gonna show you at the end an example in which we actually have one of the channels completely filled with gel. Besides that, we have a dynamic environment. So we have the fluid flow. So both channels can have this flow, which is independently controlled by our instrumentation. We can have mechanical forces. So in this cartoon, you can see those arrows. This is the direction in which we can stretch those chips. So again, trying to recapitulate the breathing motions and the peristalsis that the cells experience in the body. And we can also incorporate air-liquid interface, which is a very important condition, for example, for the lung models. And just also to give you an idea of the flavors that we have. So we have this dual channel flow, right? So we have our aluminum and vascular channel. Our flow is unidirectional. I'm gonna show you a scheme on this. So there is no recirculation on our system. The flow rate can vary from 0 to 1,000 microliters per hour. And we can have the stretching varying from 0 to 10%. So there's really lots of cultural conditions that you can apply to these chips. On top of that, we can increase the complexity of these models by adding circulating or resident immune cells, stromal cells, microbes, or hypoxia. And ultimately, we can use these chips for therapeutic testing. So all types, right? So efficacy, safety, pharmacokinetics, mechanistic studies, biomarker investigation, as well as disease modeling. So just to give you an example of how important it is to have this dynamic environment, I'm gonna show you renal tubular epithelial cells. So this picture is showing F-actin staining at a static culture. And if we expose these cells for just a couple of hours to this continuous flow, we can see a complete rearrangement of the cytoskeleton, really showing how the biology of the cells change once they are in this dynamic environment. This is our instrumentation. That's actually our Emulate system. So on the top, we have the instrumentation. On the bottom, we have the consumables. So the chips are the ones down here. This is where we see the cells. And then the chips get connected into the pods. So the pods serve as media reservoir for the cells, as well as it becomes the interface between the cells and the instrumentation. So on the top here, we have the Zoe. So the Zoe is where the magic happens. So it allows us to set up the specific flow rate and the specific stretch conditions for your experiment. And we know that, it's a consistent and robust system. So all the 12 chips are going to be experiencing the same features. And the Zoe does not have temperature control, but it goes on your normal incubator. We can fit up to 2 Zoes per incubator. And then we have the orb, which is the round one. It actually sits outside of the incubator. It provides the CO2, the power, and the vacuum for the stretching on the Zoe. And one orb can actually operate 4 Zoes, so you can actually increase your throughput by staggering the system. And to give you an idea of how the pod works, so on the pod, we have the separation between the bottom channel and the top channel, so we can treat these cells with different culture media. And we also have a separation between the fresh media, which is the one here on the inlet, as well as the spent media. So as I mentioned, our flow is unidirectional. So what happens is when we put this pod unit inside of the Zoe, there is a pressure-driven flow that takes the media from the inlet, it passes through the cells, and then it gets collected here on the outlet. So over time, we end up with this outlet accumulating effluent, which you can now collect at different time points for different types of effluent analysis. So permeability analysis, cytokines, metabolites, and so forth. In terms of imaging, just for you to have an idea, when you want to image your cells, you would just stop the flow on the instrument, and then you can remove the pods. As you can see, there is a gasket, there is a lid here, so your media is contained and sterile. During this time, the chips are transparent, which allows you to look at the cells at any time in bright field, DIC or phase contrast microscopy. The only requirement would be to have an inverted microscope that allows you to fit this pod unit on there. And once you finish your experiments, you can actually disconnect the chip from the pod. And now you can either place this chip on a microscope slide and image one by one, or we also offer these adapters that allow you to acquire image of 12 chips at once. And just to give you a little bit of an illustration of how we work. So Actually, when anybody buys an instrument, we offer a complete training. So we teach you not only how to handle the chips themselves, but also how to handle the pods and how to really adapt your research to the organs on chips. So then, it's a smooth transition. And so now we're going to move on into the paper from the Wyss Institute. And if you don't know this paper, I think you're going to find it very interesting. The title of this paper is Modeling Radiation Injury-Induced Cell Death and Countermeasure Drug Response in a Human Gutted on a Chip. So in this paper, we actually used the KECO2 intestine chip. So we seeded KECO2 cells on the top channel as our epithelium, and then HUVECs on the bottom channel for the endothelium. And we have observed before that the advantage of culturing KECO2 cells in a dynamic environment is that they form these villi-like structures in 6 days of culture, in contrast to static transwells that take 21 days for them to form. So we let these chips mature and form these villi-like structures. And then, and here's actually some characterization of it. So by DIC microscopy, we can see those three-dimensional structures. Here we can see the proper trijunctions by Z01 and the apical polarity that we actually achieve on the epithelium by staining with Villing, as well as here are some representative images of our endothelial cells, PCAM1 as a marker, the adherent junctions, as well as weak adhering. And when we expose these chips after they are mature to radiation, so the dose that we actually use here was 8 gray, because it's a dose that was shown to induce damage in both humans and animal models. And we actually observe significant cell death. Here we're showing tunnel staining at 24 hours and 48 hours. And this is the quantification of the staining. And it's actually striking how we see more damage on the endothelium than on the epithelium. And this is in terms of tunnel, because when we look at LDH, we actually see pretty much of a similar level in terms of this cell membrane damage. Now that we have a radiation model, a radiation induced damage model, we can now test countermeasure drugs. So in this paper, we use the DMOG, and we actually pre-treated with the chips for overnight with 1 millimolar of DMOG, and we see that there is a reduction of the cell death. There is a reduction as well on the LDH, both on the epithelium and endothelium cells, showing that this drug has already been used in vivo to show countermeasured effects, and we are able to recapitulate those effects on the chip. Looking at other types of phenotypes that are associated with radiation, we have ROS. So we can see here that, again, a similar trend in which we see more ROS being induced on the endothelial cells than on the epithelial cells. But again, when we treat these chips with DMOG, we suppress that oxidative stress phenotype. Moreover, DNA damage. So we know that radiation can induce double-strand breaks. And a way for us to look into permanent double-strand breaks is by looking at this foci of 53 binding protein 1. So when we're looking at cells that are healthy, we see mostly a more diffuse staining, but when the cells are damaged, we actually see these punctuate foci. And again, we see this similar trend that the endothelial cell seems to be much more affected with this DNA damage than the epithelial cells. But upon treatment with DMOG, we are able to dissolve some of those foci, and the cells are much healthier after the drug treatment. As well as permeability. So as I mentioned, because we have those two channels, we can actually do permeability analysis. And just as a summary for you guys to understand if you don't know, but we basically expose one of the channels to a fluorescent tracer. In this case, is the intestinal channel. And then we measure how much of that tracer leaks into the vasculature over time. And that is a surrogate for barrier function, right? For the epithelial and to the endothelial integrity. And what we're looking here, so we see that at 0 hours, all the chips have a very low permeability. Once we add radiation here on the black bar, we see that there is an increase in the permeability, which is already significant at 24 hours. But again, once we treat with the DMOG, we are able to rescue some of the barrier function phenotypes. showing the feasibility of testing therapeutics in these types of models. We also know that in vivo radiation is associated with villi blunting and morphological changes. So this is actually a cross-section of the chips, and it's staining with villi for us to appreciate the height of these villi. So we can see very clear that in the control, we have those very prominent villi-like structures that resemble the intestine. When we switch to radiation, there is a loss of those structures, and by treating with the drug, we can actually recapitulate, recover some of those villi-like structures. So the damage is rescued, is avoided. We can look deeper with scanning electron microscopy, and we can see that there is actually irregular and shortened microvilli when these chips are exposed to radiation, which is in agreement with the findings in vivo. And then again, by co-treating these chips, pre-treating these chips with DMOG, we rescue that phenotype, and we see again those very dense microvilli similar to the control. And also in terms of immunofluorescence, so looking here at the tight junctions, we see very nice Z01 and Z-cadherin staining when we have the healthy chips. The radiation really impair some of those formations, the continuity between cells. And again, when we're treating with the countermeasure drug, we rescue that phenotype. And finally, because we saw this trend that seemed that the endothelium was having a more severe effect, we actually analyzed what is the importance of the endothelium in this model. So we actually made chips with only KECO2 and then KECO2 and HUVEC, and we exposed them to radiation. Interestingly, we only see the villi blunting when the endothelial cells are present, as you can see here. So it really suggests that the endothelial cells are signaling the damage to the epithelial cells. And when we look at the permeability, we see also a very similar effect. So here is at 0 hours, right? So no really difference between. So once we add the radiation, we can see that only when we have the endothelial cells, we can see that effect, right? So here at 24 hours, we already have significancy when we have the endothelium, but the chips without the endothelial cells have no difference at 24 hours and at 48 hours. So it seems like if the epithelial cells are alone, they have a delayed response. Only at 72 hours, we actually observe this permeability being significant, which really suggests that there is a crosstalk between the endothelium and the epithelial cells, and the damage to the epithelial cells is actually downstream to the endothelium. And also in terms of mucin, so again, if we look at the chips that have the endothelium, this is the healthy one, and we can see here the goblet cells is stained with mucin. When we irradiate those chips, there is a loss of both the tight junctions and the mucin, but if those chips do not have an endothelium, we do not see that phenotype. So I hope you guys appreciated this data, or at least it gives you some ideas for possible projects on the intestine or in other organ models. But the idea is that, you know, we can actually study radiation now with a much more human relevant model that can have, you know, different cell types, and that allows you to do therapeutic testing in a much short period of time than you would do with animal models. The KECO2 is one of the first models that we worked on, but if we look at the timeline of the company, our first product was actually the liver chip. So this is the first product that we released on the market with validated cells. And the main focus of that application was drug absorption and metabolism, as well as safety and toxicity. And in 2019, we launched 2 new organ models, the small intestine chip, and that's with biopsy-derived organoids, as well as a primary proximal tubule kidney chip. Again, those two models were focused on these drug metabolism and safety toxicity applications. Last year, with the pandemic, we actually shifted our focus into infectious diseases and lung models. And now, for the next upcoming years, we have a lot of exciting things coming up. We have applications for inflammation, immunology, immune cell recruitment, microbiome, and the 2 models that we are launching this year are the brain chip and the colon chip. And we're actually going to talk about a lot of those. So these are the models that we're going to review today. So the liver, the colon, the brain, alveolus, airway, and lymph nodes. So hang there with me. I hope you enjoy this. So let's start with the liver chip. So the liver chip, we started this collaboration with pharma partners as well as with the FDA and the DARPA grant. And we actually started with a very simple model of a co-culture in which we had hepatocytes and endothelial cells. We can actually increase the complexity of this model and make it into a tri-culture by adding Kupffer cells, which are liver macrophages, and even further create a quad culture in which we add stellate cells. So just to give you an idea of one of the advantages of our liver chip is that we can have a much longer culture than you could have in transwells or in 2Gs. So similar to what Molly showed you before. So basically it takes us about 7 days to seed, to prepare the chip, to seed the cells on the chip and to have the culture stable. And then we actually can start our experiment and we can treat these chips for 14 days. So, if you have a drug that has a very mild effect, but it's a long-term effect, now with the chip, you can actually investigate that because you can keep this culture for such a long time. And for this project, not only were we interested in the human liver chip, but also on other species liver chip, because we wanted to see if we can use these chips to make this in vitro to in vivo correlation, right? Because if we want to say that the chips are a good surrogate to human biology, and that's because of the microdynamic environment, that it would be the same for the dog, for the rat, and any other species, right? And since those are the main preclinical species that we used, we actually focus on those, on this liver chip work, so then we could see the effect of drugs, from the preclinic into the humans. So to start with, I wanted to show you some representative images. So we have on the top bright field images of the hepatocytes, and on the liver chip, we can see that typical hepatocyte morphology, with the hexagonal binucleation, and this is at 14 days of culture. In comparison, when we look at the plate, you can barely see the hepatocytes, right? So you do not have the same viability and longevity of the culture in the plates as you have when they are in this dynamic environment. Moreover, in terms of functionality, we can flow the chips with CGFDA. This is basically a dye that gets pumped out of the hepatocytes when they are viable. And by doing that, they actually stain the bilicanaliculine network. So what we see is that when we have these chips cultured when we have the hepatocytes cultured in the chip, we can have a much better definition of these bilicanaliculine networks compared to the plates. Moreover, if we look at functionality of these hepatocytes in terms of albumin and urea secretion, we also see a much better secretion levels on the chip when we compare it to the plates. So here I'm just showing the human and the rat, but that's the same for the dog. And we see that is a significant increase the level of secretion, right? We barely see any secretion or functionality on the plate throughout these 14 days of culture. Very important for liver work is the activity of cytochrome P450 enzymes. So we tested many prototypical probe substrates to test for the different isoforms. And again, in the human and the rat and the dog, and the trend is always the same. So here we are comparing, the dashed bar is the fresh hepatocytes. This is the golden standard for toxicity studies. And then we are comparing that with the hepatocytes on the liver chip in gray and the hepatocytes on the plate in black. And for the most part, we see that the chip has a much better CYP activity than the plate. And in some cases, it's even higher than the freshly isolated hepatocytes. And again, it allows you to study those effects for a much longer period of time. In terms of transcriptomics, we also see big differences when we compare the plate with the chip. So specifically here, we are looking at some proteins that are very important for transporter or enzyme functions. Again, depending on your application, this really justifies the use of the liver chip versus the plate, because if you don't have the activity of your specific transporters, then you're not going to be able to investigate your biological question. To characterize the extra cell types on the chip, here I'm showing on the top the Kupffer cell characterization. We are able to see phagocytosis of these cells. So we can introduce, for example, LPS as an insult, and then the cells will be activated, and we can actually add latex fluorescent beads to be able to see the phagocytosis happening real time. And we can also stain with CD68, which is a marker for activated Kupffer cells. Another way to look at functionality is by looking at the secretion of inflammatory cytokines. So it's well known that Kupffer cells are very involved in the inflammation. So what we're looking here is in blue, we have the co-culture, so just hepatocytes and L-sacs. And then we have the tree culture with two different amounts of Kupffer cells. And we can see that the secretion varies, right? So if we have more Kupffer cells, we also have more secretion of the cytokines, really showing that that's a direct effect of having the functional Kupffer cells on the system. But you know, again, depending on the cytokine you're working with, you don't necessarily need a triculture, right? So IL-6, for example, is one of the few cytokines that is induced on L-sacs as well. So that's why we can actually see the IL-6 secretion already in the co-culture model. We also characterize our stellate cells. So stellate cells, they are mostly quiescent, but then when they get activated with some sort of injury, they basically they become much more fibroblastic, and they become positive for alpha smooth muscle actin. So we actually, in this case, we activated the chips with TGF beta for 48 hours, and we are able to see this very nice phenotype, morphological change, as well as protein expression of the stellate cells. Moving on to some data. So with the liver chip, we expect to address two gaps. The first one is to question the toxicity that we see in animal models, right? Is it really true that in humans? So this is an example from J&J of a compound that it was actually being used in rats. And after 2 weeks of treatment, that compound actually induced fibrosis in the liver. So this compound was discontinued, for further testing in no rodent species. So we made a human and a rat liver chip, and we tested this compound to see what would be the effect in humans. So as expected, on the rat chip, we do see that significant increase in the stellate cell activation. However, on the human chip, we don't see such response. If we quantify those positive cells, we can see a very nice dose-dependent increase on the stellate cell activation when we expose the rat chips to this drug. But in the human chips, we don't see that. So even here at the highest dose of this compound, we still don't see as much damage as we see on the rat, really highlighting those species-specific differences. And also in terms of functionality. So not only the stellate cells are activated, but we also see that in the rat, there is a decrease in the albumin. So there is a decrease in hepatocyte function already at 4 days of treatment of this compound. And in the human chips, even when we treat these chips for 14 days at the highest concentration, we still don't see that decrease in albumin. So the message here is really that, this is a compound that perhaps could have been used in humans, but the development was stopped because we saw toxicity in animals. In addition, we have another gap, and this is definitely the most important gap, right? This is when we don't see the toxicity in animal models and we think we're good to go, but then we see the worst happenings in clinical trials. So this is the example of filarudin, which is an antiviral nucleoside analog that was actually discontinued in phase 2 clinical trials because it caused liver failure and death in 5 out of 15 patients. And the phenotype was caused by steatosis, which is an accumulation of lipid within the hepatocytes. And so what we're looking here, so Nile red is one staining that we can use to stain the lipids. And we see that even at 30 micromolar, there is no significant lipid accumulation on the rat liver. However, when we look at human liver chips, already at 1 micromolar dose, there is already a significant increase in steatosis. And on the side here, we can see the quantification of these images. We can also look at albumin again to take a look at the function of the hepatocytes. And we can see a very nice dose-dependent decrease in function in the human hepatocytes, which was not really predicted in rats at any of those doses. And we can look at different other markers, right? So miR-122, alpha GST, and keratin 18, they are associated with responses to oxidative stress, and they all showed that a toxic response in the human liver chip. So here highlighting the importance of having a more human relevant model when testing the toxicity of drugs. We're gonna move on into our colon intestine chip. So in this model, we are using, so actually, let me recap for a minute. So on the liver model, all our cells are primary human cells, those results that I showed you. In this case, we are using organoids, patient-derived organoids as the source for the epithelium. And basically the way it works is, we get the biopsy, you expand those organoids in 24 well plates in a matrigel culture, and then we can actually fragment those organoids and seed them into the top channel, and they will form a monolayer as well as differentiate into the different cell types. So this is just a representative images for us to have an idea. So this is how the organoids look on the matrigel culture, and this is how they look after they were seated on the chips and they are in the flow culture. On the bottom channel, we use human colonic microvascular endothelial cells. And the cool thing about this model is that you can actually get very specific endothelial cells. So here we are using colonic endothelial cells. If you're doing a small intestine model, you can actually use a small intestine endothelial cells, and you can have a very specific and matched biology for very specific questions. And again, just to give you an idea of the timeline of these experiments, so in the case of the organoids, we can actually keep these chips for a little bit less time because there is a big turnover on the organoids, and we cannot keep them very viable for much more than 8 days in culture. But basically, we still have 4 days in which you can perform, drug treatments, microbiome experiments, and so forth. So as a characterization of our colon chip, so as I mentioned, so we seed the cells on the chip and then we let them differentiate in this dynamic environment of flow and stretch. So for the liver, we don't use the stretch because that's not physiologically relevant, but here on the intestine it is. So these cells are exposed to a stretch starting at 2%, and then we increase to 10%. So during this culture, the cells differentiate, and we are able to see all the main intestinal types that we see on the colon. So we have here the absorptive enterocytes, which are stained with villi. We have goblet cells, which are mucus producing cells. And we also have the enteroendocrine cells. And we see them, there is also a donor to donor variability in the amount of those cells, because of course you're working with patient-derived samples. In this project, we also tested for the contribution of the endothelium co-culture. So what we're seeing here is the staining for effectin, ZO1, and E-cadherin. And interestingly, when we don't have the endothelium, there is an impairment of the formation of this tight junction. So either we see a decreased expression of those proteins, or we see a complete relocalization of those proteins from the membrane into the cytoplasm when these organoids are being cultured by themselves and don't have the extra signaling that endothelial cells provide. As a consequence of this impairment on the tight junction formation, we also see differences in permeability. So this is a very similar assay than the one that I mentioned before. So the black line here is the colon chips, just monoculture, and then the blue ones are the ones with the endothelial cells. And we can see that from the very beginning, the ones with the endothelial cells, they already have a low permeability that becomes even lower once the chips mature. But when we don't have the endothelial cells, it's like we have a delayed process of maturation of the cells as well as the barrier function of these chips. The culture with endothelial cells even interferes with the polarization of the epithelium. So here we are looking at the example of the sodium potassium ATPase, which is a basolateral marker. And we have the DRA transporter, which is an apical marker. And we can see here the polarization, the correct polarization, similar to in vivo when we do have the co-culture and this polarization is lost when we only have the organoids on the chip. So again, just to emphasize the importance of having very complex models with different cell types to really be able to recapitulate what we see in vivo. And in terms of gene expression, we see that our chips are closer to the human colon than organoids. So what we're looking in this plot, it's called the transcriptomic signature distance. This is a metric that we published last year, and it's basically comparing the gene expression patterns between different data sets. And the distance is basically the dissimilarity, right? So what we're looking here is that there is a significant difference between the same organoids cultured in Matrigel or cultured on the chip. And because now these organoids are cultured on the chip, there is a shift in gene expression towards a more human-like pattern. And that's why we don't see a significant difference between the human colon and the organoids grown on chips at day 8. So again, showing that we do have, by providing the cells with this dynamic environment, we are making them be more close to the human biology. Now that we have this colon chip, we can use it to study inflammation, for example, because there are many GI diseases out there that are very related to chronic inflammation, such as ulcerative colitis and Crohn's disease and IBD. So this is one representative protein. On our paper, we have tested many tight junction proteins, but basically we expose these chips to interferon gamma as a very standard cytokine to study GI inflammation. We did that for 48 hours at increasing doses. And what we see is that there is definitely changes in morphology, right? So there's changes in cell size, there is changes on the distribution of these proteins throughout the tight junctions, and that's very related to permeability again. So we have tested these with 3 different donors right now, and all of them respond the same. So we see a dose-dependent increase of permeability, both right with the dose as well as over time, and in which, so the barrier, the epithelial barrier is being disrupted. And that is in part because of this deficiency on the tight junctions, but also due to apoptosis. So if we look at the full channel of the chip, this is what we're looking here is basically the DAPI coverage of the chip. So we have our vehicle as being full confluent and healthy, but then as we add interferon gamma, we see that there is a decrease in the coverage of those chips, which means that there is some cell death occurring. And if we look at the caspase signaling, we actually were able to confirm that it was caspase-induced apoptosis. And this is very important because interferon gamma is a cytokine that has been widely studied in the literature, and it's known that the mechanism for to cause leaky gut phenotypes is both towards deficiency of the tight junctions as well as apoptosis. And we are able to recapitulate both of those phenomena on the chip. And once we have the inflammation model, now we can test for anti-inflammatory drugs. So we used tofacitinib, which is an approved drug for ulcerative colitis. We are using the dose of 20 micromolar here. And basically we were treating with tofacitinib for 48 hours and then interferon gamma for 36 hours. And we can see that there is a rescue on the phenotype. We can see that these cells here that were treated with both the interferon gamma and the tofacitinib, they look very much more like the control. They don't look as damaged or altered as the ones that were just inflamed. We also see a rescue in permeability. So the pink line shows the barrier dysfunction over time that we see in these colon chips in the presence of interferon gamma. And the blue lights are either the tofacitinib by itself or the tofacitinib with interferon gamma, right? So we really rescue that barrier dysfunction that occur due to inflammation. And in terms of cytokine release, so as I mentioned before, we can collect the effluent from these chips and perform cytokine analysis. So we can see that interferon gamma increases, right? Comparing here to the vehicle, increase the level of several cytokines. We are only showing 3 here. ICOM-1 as one cytokine that is mostly secreted apically, and VCOM-1 and IL-6, which are mostly secreted basally through the vascular channel. And in all the cases, once we add the co-treatment with the anti-inflammatory drug, there is a reduction on the level of these inflammatory cytokines. So again, showing that we can use these models to study the healthy phenotypes, induce insults such as inflammation, and now use drugs to be able to test the efficacy of those drugs towards certain phenotypes. Let's move on into our brain chip. On our brain chip, we have all the key players for recapitulating the interactions between the central nervous system and the vasculature. So we can actually think about this model as two units or two modules. We have the blood-brain barrier here composed by the astrocytes and pericytes, which would be on the top channel, and then the microvascular endothelial cells, which would be on the bottom channel. And we can add complexity to create a neurovascular unit by adding now neurons and microglia. These neurons, you can add any neurons that you would like. In this particular case, we are actually adding cortical neurons, and we are able to show that both glutamatergic and GABAergic neurons are present in this culture. And to give you an idea of the timeline, so these experiments are also shorter experiments. So we are able to stabilize this chip much quicker. So in 3 days, we can already expose these chips to compounds. And then we have a window of 4 days in which we can experiment on this model. This is live calcium imaging of the top channel showing spontaneous neuronal activity on the chips, showing that the neurons are functional. So this is to show the variety of the readouts, right? So we can do imaging readouts, but we can also do effluent readouts to be assessing the functionality of the neurons. So in this case, we are looking at glutamate being secreted into the effluent because we know we have glutamatergic neurons, and we see a consistent secretion of glutamate throughout the culture. Similar to the plots that I showed you before for transcriptomic analysis, what we're comparing here is all the same cells that we have on the brain chip, but cultured in trans wells. This is the pink boxes, and then we have the brain chip here in blue, and the human brain in gray. And in this case, we actually had 2 time points, day 5 and day 7. And in both of them, we see the same trend, right? So we do see a significant difference between the human brain and the cells just cultured in a simplistic 2D model. But when we culture those cells on a dynamic environment, now we become much closer to the in vivo gene expression pattern. Again, emphasizing the importance of having this dynamic environment for the cells in a more complex environment. We can also induce inflammation on the brain chip, and that's important because a lot of neurodegenerative diseases are associated with neuroinflammation. So for this particular model, we actually use TNF as our cytokine to model inflammation. And after 48 hours of TNF exposure, we observe astrogliosis, which is the increase in astrocytes, here shown by GFAP positive cells. We also see microglia activation by staining with CD68. And there is a morphological change really on the microglia once we add TNF alpha. So they go from this more ramified stage into a more amoeboid stage. And again, we can also do live imaging and add fluorescent latex beads to be able to see the phagocytosis and the functionality of this microglia. And if you are going to study inflammation, that's when the microglia is really important, right? So as we can see in this plot here, we are comparing chips that were treated with TNF or not in the presence of microglia or not. And for the most part, what we see is that there is an enhancement in the levels of secretion of the cytokines when we have the microglia than when we don't. We're looking here at interferon gamma and IL-1 beta, but specifically to IL-1/6, we don't really see any induction of IL-6 when we don't have microglia. So again, depending on the specific cytokines and the specific phenotypes that you were interested on, having the microglia can really be important for defining those mechanisms. And we can also do permeability analysis on the brain chips. Here we are showing after the TNF, right? So at 24 hours, we see a trend for increased barrier dysfunction, but at 48 hours, that becomes very apparent. And now we can use these models for modeling different diseases. We also have a paper on bioRxiv right now that instead of using cortical neurons is using dopaminergic neurons. And you can model things such as, the alpha-synuclein pathologies. So there are many different types of injuries that we can add to these chips, and then, many different readouts of functionality that can recapitulate in vivo responses. Now we're going to go into the lung world. We're going to start with the alveolus lung chip. And for this chip, we are using primary cells from commercial sources. When you get the cells actually from the vendor, they come as a culture of mostly alveolar type 2 cells. So the alveolus has two main types of cells, the type 1 and the type 2. The type 2 are mainly involved in homeostasis, and they are actually the progenitors of the AT1, which are the ones that are actually perform the alveolar function of gas exchange. And by culturing these cells on the chip in the dynamic flow in the presence of endothelial cells, by day 14th, we can see that there is a differentiation, and we can see basically a mixed population of both of those alveolar cell types, which is very important because if anybody tried to culture the cells before in 2D, you know that it's very easy to end up with a culture of just AT2, and you're not really going to be able to access the alveolar specific functions on those models. We confirmed that we got proper tight junction formation both on the epithelium and endothelial cells. And we looked at the gene expression profile of these chips, because now here we also have the peristalsis, right? Here we also have the stretching as a mechanical force that is very important for the lung function. So we really wanted to see which changes are occurring throughout these 14 days of culture in these alveolar cells. And we see that there are genes related to differentiation. So here we are looking at genes that are markers of alveolar type 1 cells, so aquaportin 5 and podoplenin. They are increasing during the time of culture, and as well as markers of alveolar type 2, which are also maintained. These two are markers are related to surfactant function. We also see changes in IO transporters, and that's important for fluid clearance and maintenance of the air-liquid interface. We also see changes in defense response. So ACE is the receptor for SARS-CoV virus, and we can see that it's actually increasing expression upon, over time in culture, as well as chemokine CCL5. And not only the epithelial cells undergo changes in this dynamic environment, but also the endothelial cells. We saw the upregulation of many Wnt ligands that are important for this crosstalk between endothelial and epithelial cells during development. And if we do an analysis, a principal component analysis, just to see how those samples distribute in the plane, we can see that the transwell is completely off, right? So we do have here a trend that, at day 8 and day 4, these cells are much more similar, so they are already differentiated. But the transwells, it's a completely different pattern, right? So two completely different models. We are not sure if this very simplistic model is going to show us what we want in terms of the complex human biology. And here we're going to focus the presentation today into virus infection. So we wanted to check for the feasibility of our models for influenza A infection. We used, as you can see here, different strains of the virus, and we observed that while the H3N2 and N5N1 could infect the alveolar cells, the H1N1 did not really. And this is consistent with findings that these viruses actually preferentially infect the airway and not really the alveolus. So not only we see the susceptibility, but we also see specificity of response in this model. One of the main characteristics of the innate immune response to virus is the production of cytokines. And we are able to see the production of many cytokines on our chip upon viral infection. And when we look closer to the type of response that we're actually getting a response that is related to the interferon type 3. And this is similar to what we see in vivo. Some transwell studies have actually found that the response was more related to interferon 1 and interferon beta. And that's really not what happens in vivo. So this suggests to us that, this by culturing these alveolar cells on the chip, in terms of viral infection, we are activating the pathways that would be activated in vivo. The endothelial cells also undergo changes when we infect these chips apically with the virus. So we see markers of inflammation. So we see here upregulation of ICOM-1, as well as upregulation of the secretion of TNF alpha. And because we know that in vivo leukocytes are recruited to the site of infection, we tried to test that in our chip. So we isolated PBMCs and we stained them prior to introduce them into the vascular channel of these chips, the control and the ones infected with virus. And we can see that because that endothelium is activated upon the virus infection, we see that there is a recruitment, a significant increased amount of PBMCs that are recruited to the endothelium and actually bind to the endothelium. And because we had this idea that it seems like the chips, when they are in this more dynamic environment, they have these defense response, right? So we saw that interferon response and the cytokines. We asked the question, but what if there is no briefing motions, right? So we prepared the chips and then we infected them being exposed to the mechanical forces or not. And very interestingly, we saw that when we have the mechanical deformation, we actually have less virus infection. So here we are looking at immunofluorescence for virus protein, but also when we look in terms of viral titers, we see that there is less proliferation of the virus and, and less release from damaged cells. As expected, we also see less cytokine production when these chips are under this mechanical deformation. When we do this comparison between the mechanically deformed and the static, we see that there is a lot of genes that are related to inflammation that are upregulated on the mechanically deformed chips. And there is, we also see on that response, so there we can see that there is activation of these mechanically stimulated pathways. So that shows in the RNA sequencing, as well as these defense responses pathways. And at the same time, we see a suppression of cell division and cell cycle proteins. And again, this is just another way of looking at the data here, looking at those interferon stimulated genes, which are very much upregulated at day 14 after this dynamic culture, which we do not see either at the day 0, right, or at the transwells. So again, if we want to study virus, we definitely want a human system because that's how the specificity is going to play. But now we do have indications that really the mechanical deformations and then the flow can play a big role on the type of infection that you're going to see and if that's going to recapitulate the human infection or not. Moving on to the airway lung chip, we are going to start with the small airway. So as I mentioned before, depending on the cell type that you use, you can build a different model. So here we're looking at the small airway in which we are able to observe the differentiation of the epithelium into the very important cell types of the airway. So the club cells, the ciliated cells, and the goblet cells. Not only we see those cells present, but we also see them present at similar percentages that we would see in humans. So the one in blue here is what we see in vivo, and the one in pink is what we see on the chips. And we can see that we are very close to the human biology. We can also probe the functionality of these cells, for example, by looking at the cilia beating once these cells are differentiated, as well as the movement of mucus. And again, if we actually extract values from these readouts, we can see that we are very close to the human biology. So we're looking here at cilia beating frequency, the cilia length, as well as the velocity of this mucociliary clearance. So all these features of the airway chip are very resembling the human airway in vivo. And we looked for the expression of these proteins, serine proteases that are important for viral infections, as well as the ACE2 protein. And we can see that here we are showing the pre-differentiation and post-differentiation of these cells. So after the full differentiation on the chip, we see a very significant increase in the expression of these proteins, which makes this model suitable for viral infection. But also when we compare these models to, for example, MDCK models, we see a much higher expression of these receptors. And as a proof of concept, we infected these chips with H1N1, and we are able to see the cytopathic effects that we expect, right? So damage to the epithelium, loss of tight junctions, loss of ciliated cells, and even loss of the vascular integrity as well. And we can do also a bronchial airway model by using the cells from the large airway. And again, we infected these chips with virus, different specific strains. We can see that they all grow in the chip up to 72 hours. And we also wanted to increase complexity here and add neutrophils and see how by the addition of neutrophils would take care of these infections. So what we're seeing here is this is the addition of the neutrophils. We see similar to the other experiments that the neutrophils within minutes, they actually bind to the endothelium. And then within hours, they migrate into the apical lung channel. So this is actually, these pictures are on the airway channel. And basically what happens is the neutrophils transmigrate, they target the cells that are positive for the virus protein, and they make these cells coalesce into these clusters, and they actually end up inducing cell death. The quantification of some of those studies show us that there is a difference between the virus strains. So H3N2 seems to be much more severe, the infection that it induces. And consequently, we actually see a higher recruitment of neutrophils to the top channel when we do have this specific strain. But in general, the addition of the neutrophils actually reduce the virus titers of these chips, which is exactly what we would expect from a healthy individual, right? That the immune system would be able to defend them. And we see that in terms of cytokine production, the addition of the neutrophils actually enhance that cytokine production, which makes sense. And again, we can see the difference between the different strains on the activation of the inflammation on these chips. And we can also use these chips for testing possible prophylactics or therapeutics. This is using the SARS-CoV pseudovirus. So this is the virus is expressing the spike protein, and we were able to see the reduction of the viral titers in some of the drugs tested on this paper. So showing that we can model both complex responses related to the immune system, but also using for efficacy testing upon viral infection. And finally, our lymph node chip. So this is a chip, I must say that we, comparing to all the other ones, we haven't done as much work at Emulate as the VIS. So this is mainly work from the VIS Institute, but it's also a very important model, right? So there is very limited in vitro and animal models that are capable to emulate the human lymph node physiology, as well as to be able to accurately predict the safety and efficacy of vaccines and immunotherapies. And that's why a model such as the lymph node, it's so important. Also, I wanted to show this model because it has a very different configuration from all the other models that I showed. So if we can see here in this schematic, we actually have our bottom channel being filled with a gel. And in this case, we can have the B cells and the T cells embedded in this gel. You can also add dendritic cells if you're trying to add complexity to your system. And then we have that porous membrane. And on the top channel is where we have our media. So so the cells are getting the nutrients from the media on the top channel, as well as excreting everything into the media in there. And in this is also a pre-publication in bioRxiv. And what we were able to show with this lymph node chip is that we are able to recapitulate in vivo-like follicle self-assembly, antibody production, antibody class switching, plasma cell differentiation, and in vivo-like response to influenza vaccination. So very briefly here, I'm gonna show you guys some pictures. So we are able to see the self-assembly of T and B lymphocytes into these lymphoid follicles on the chip. Only under flow, we see this self-assembly already at day 4. We also see this realignment of the fibrils. And we see that this follicle formation, when the chips are cultured when the cells are cultured under flow, we see that there is an increase both in the follicle number as well as in the follicle volume when compared to static cultures. We can also see class switching of B cells. So basically, when B cells get in touch with an antigen, then they can actually undergo class switching, so then they can start producing IgG. So in this particular case, we actually use the SAC, which is a bacterial antigen, and we can see that these B cells now are producing the IgG against it on the chip environment. We also see plasma cell differentiation against this bacterial antigen. So here we are seeing this, the marker is the CD138. It's the marker for the plasma cells. And we see that they are enriched within the lymphoid follicles, which is what is expected from in vivo findings. And if we use a vaccine, we actually use the Fluzone vaccine in this chip to see if we could mimic the immunization response. And we did. So we actually used different donors, right? So the B and T cells were from different individuals. And at day 9, after applying the vaccine to the chip, we can see that we have a range of response, right? Exactly like you would see in vivo in humans. You would have your low responders as well as your high responders. And when we look at this, is all compared to a tonsil control. And when we look at this antibody production on the chip, we can also see these two groups being very evident, but we see a consistent antibody production over time on the chip. So I hope I was able to show you guys today the variety of things that we can do with these chips. Lots of applications, lots of models, lots of different ways to set up the models into the chips. And we can really use them for many, many applications. So basic research, we can understand very basic processes of healthy tissue, as well as start investigating diseased organ models if you have the, diseased cells from diseased individuals. We can study the interactions between different cell types, those being, epithelial and endothelial cells, but we can also add the immune cells. We can study disease pathogenesis in multiple organs, right? So COVID, for example, we don't necessarily need to study COVID only on the lung. We know that other organs are also susceptible to that. So you could potentially use different models to study the same pathogen. We can also use those chips for drug discovery and development, both in terms of efficacy and safety, but also in terms of mechanism of action, as well as more clinical applications in terms of discovering and validating biomarkers or exploring variations in different patient populations. And I would like to close with the discussion that, I know that a lot of us have been very used to work with animal models. That is the easiest, that is the golden standard, and we already know how to work with them. But that's really not the answer, right? So we really have to move forward with more human relevant models and which are also more humane and much more predictive of the human response. And hopefully by doing that, we're gonna get a better understanding of human biology, diseases, response to injury and countermeasure drugs, as well as toxicity to drugs and other chemicals. And I really appreciate your attention today. Thank you so much for being here with me, and I'm happy to take any questions.
Graciela, the first question is is from Debtor. How difficult is to prepare the samples?
So I guess it would depend on which sample we're talking about, right? So for example, if you're working with iPSC-derived cells, you're definitely there is a time, like a couple of weeks, that you're gonna have to differentiate those cells before you add them into the chip. Same thing with organoids, you're gonna have to expand them prior to the chip. But on the liver chip, we will provide you with the cryopreserved hepatocytes, and they literally go from the liquid nitrogen into the chip. So it really depends on the model and the cell sourcing that you're using.
So the next question is, how representatives are the results to in vivo models?
So as I mentioned, right, so most of our studies are actually with human cells because we're really trying to do that surrogate to the human biology. For the liver, we actually did the preclinical species, so we were able to compare directly, for the rat, for example, the liver, the liver animal and the liver chip. So from our findings, it seems that the biology is much better recapitulated. But I have to say that for all the organ models, all the other ones, we haven't really done the other species because we've been focusing on the humans.
Next question from Paula. When co-culturing epi and endothelial cells, how do you choose an ECM?
That's a great question. So in some cases, there's lots of literature that at least you can refer yourself to, right, as a starting point. But you can just do test really to see, because literature sometimes it's not really that precise. But basically, you would have to have some readouts for functionality, right? So you would test different ECMs, for example, and then you would test, okay, are the markers of my endothelial cells present? Are the markers of my epithelial cells? Is the function of my epithelial cells changing if I change the ECM? So you should definitely look on the literature, but also perform some tests so then you can really evaluate yourself how that's changing the functionality of your cells.
How many cells are usually growing on a chip area? What is the range with the different cell cultures?
So it's from 50,000 to 200,000, depending on the model. Because, for example, the liver is going to be terminally differentiated, so you're going to have fewer cells. But on the intestine, you have proliferating cells, so you actually have much more material to work with.
And I have seen a lot of questions from Pankaj Singh. Let me read one. Did Emulate sponsor all these experiments?
No, actually, some of those experiments are actually from the Wyss Institute. They are not from Emulate. And I can actually share, so I could share all the papers that I went through today, so then everybody can have access to the full data set.
Thank you, Graciela. And I am sure if more questions are there, Cassandra can forward that to you, and then you can contact them directly by email. And if any of the attendees have any further question, I'm sure they can contact Graciela. So she will give
Absolutely.
input.
And have a wonderful evening, everyone.
Thank you.
Thank you so
Have
much,
a good
everyone.
evening.
Bye-bye.
Thank you.